with a focus on the pathogen side. And as you know, the powdery mildew is it's probably not the most important pathogen globally, but according to this, let me just get the laser pointer. According to this paper of Savary et al., it is one of the 10 most relevant diseases of wheat globally. And as you certainly are aware of, the development on the genomic side on wheat, the reference quality genome sequences that are available now, that has really transformed the field of resistance biology in wheat. And we first had the Chinese Spring reference genome sequence, and then there was the 10+ genome project where a number of genomes have become available. Now, similarly, on the pathogen side, specifically for powdery mildew, we now have very high quality genomes available, and the genome is about 150 megabases. So for a fungal genome, it's rather large, similar maybe to the rust genomes. And we have more than 10 isolates where we have now a reference genome quality, and mostly these are chromosomes from telomere to telomere. And we have more than 400 isolates with short read sequences. And this is a sort of the molecular knowledge and the data that we use for the work that I will present in my seminar. Now, this just gives a few, some statistics on the pangenome. Here are the isolates that we have sequenced, and you can see this is not only wheat powdery mildew, it's also powdery mildew of triticale and rye. And concerning wheat powdery mildew, which is here in pink, there are isolates here from Iran, from China, from Japan, United States, Argentina. So we tried with this pangenome analysis really to cover the broad diversity of powdery mildew of wheat and closely related species. And you can see here that basically this is always coming to 11 chromosomes for most of the genomes. Now, in terms of diversity, we have a global population assembled in our group, and in total there are more than 400 isolates that are sequenced with Illumina technology, and this includes our own collection, but then there is also a very good collection from Christina Kauger's group, and this provides really a huge data set for example, for GWAS studies that I will describe in a moment. And here you see a diversity analysis from a paper we published 2 years ago, which shows clearly a geographical subdivision of the mildew isolates that we have studied. Now, on the host side, there are more than 70 powdery mildew resistance genes described, genetically analyzed and described in the wheat gene catalog. And of course, these genes are very important sources for resistance breeding. Not all of them have been molecularly isolated, but there is a growing number of powdery mildew resistance genes from wheat which are molecularly known, and these are here listed in green. That's from a paper of Peter Drakatos last year, and this is already outdated because more and more genes are cloned, but I use this summary here, and you can see these are the cloned genes, and they are mostly encoding NLR, typical NLR immune receptors, but there is a growing number of exceptions. 5 of the 17 cloned BM genes last year were non-NLRs, and I will come back to that at the end of my presentation. So these are mildew resistance genes which encode NLR immune receptors, and they all confer very specific resistance to mildew. And of course, we ask the question, what do they recognize in the pathogen? Because we wanted to better understand at the molecular level what the interaction, the molecular basis of the interaction of wheat and powdery mildew. And here you can see here in green the host site. Here are a few of the genes that we isolated in our group, and we have now for most of them, actually for all of them, but this one is not yet published. We have the corresponding recognized AVR genes defined, and this was basically the work that we did in the last about nine or 10 years. And I want to describe now a little bit more what we have learned on this specific interaction, both for the wheat genes, but I will then also describe work on the two PM8 and PM17 resistance genes derived from rye. This was introgressed on chromosome segments into the wheat genome. And we study these interactions mostly at the functional level in the Nicotiana-bentamiana system. We can co-infiltrate the resistance, the clone resistance gene, and the corresponding AVR. And in case of recognition, you can see this hypersensitive response, which is very nice. It shows that this recognition and signal transduction mechanism is conserved. Even in a dicot species, you just add a mildew and the wheat gene and you get the same HR that you observe in wheat. We used a number of different strategies to isolate the molecular, at the molecular level, the AVRs. We used QTL mapping, map-based cloning, genetic approaches, then GWAS studies in the large populations that I just mentioned. We used a benchmarking approach to identify the best candidate genes based on some parameters and then functionally tested them. And what I will discuss mostly today is mutagenesis and the new assay that we have developed, which is called aberrance gene depletion assay. And I want to describe that in a minute in some more detail. What we have learned about these cloned AVR genes, which some of them you see here, when you look at the expression after 48 hours of infection, and you just plot the number of effector genes and effector candidate genes and the expression level. So you can see that basically all the functional AVRs are in the top 20% of all candidate effector genes. And this seems to be one of the important characteristics for functional AVR genes, that they are quite highly expressed. And they all encode small proteins of around 100 plus amino acids. They have, of course, a signal peptide, and they have this YFC, Y or FXC motif here in the protein sequence. So we know now what makes a good candidate gene, and that helps, of course, in the benchmarking approach. But as I will show you later, there is at least one exception, and there is probably more. So this is not the complete answer to the identification of AVR genes. We don't know the structure of many AVR genes, but the group of Paul Schulze-Leffert has studied this in a paper last year, and these are actually 4 aberrance proteins from barley powdery mildew, not from wheat powdery mildew, but from barley powdery mildew. There's only one from wheat powdery mildew that we contributed. This is AVR PM2. And you can see that these structures, which were experimentally derived, are all very, very similar, despite a large sequence divergence at the primary sequence level. So they have all this typical RNase fold structure that has been described before, and that seems to be one of the hallmarks of this wheat and barley powdery mildew aberrance proteins. Now, specifically in our work, we have been working a lot on one specific powdery mildew resistance gene. This is PM3. And PM3 exists in more than 15 functional alleles, and we have mostly focused on these alleles which are shown here. And what you see here is the overall coiled NBS LRR domain structure, and then here you see in colored bars, these are amino acid polymorphisms between the different NLR immune receptors encoded by the different alleles. But it's an allelic series of resistance genes, and there is an ancestral gene which is actually a susceptible variant, and this is the reference to, and in comparison to this susceptible reference, we indicate here which amino acid are polymorphic. And we have isolated for a number of these allelic immune receptor variants the corresponding AVR genes, which is indicated here. And as you can see here, there are two pairs of alleles which recognize the same AVRs, and I want to discuss that in a minute in more detail. So what we have learned is that in contrast to the corresponding NLR allelic series, the corresponding AVR PM3s do not form an allelic series. So we expect it may be also on the pathogen side an allelic series of AVRs. That's not the case. The different AVRs encode sequence unrelated predicted RNase fold containing small proteins that are highly expressed, but sequence unrelated. And we found that PM3AF recognized the same aberrance gene product, and PMB and 3B and C also. And we have then also isolated AVR PM3D, and we have studied the orthologous genes in rye. PM8 and PM17 are orthologs of PM3 in wheat. They are allelic in rye, so we have a nice comparison of closely related species which had an independent evolution in the last few million years of these immune receptors. So to also go a bit into these rye resistance genes. You know that rye is a source for resistance breeding in wheat, and in general it's assumed that resistance genes from rye should be more durable because the pathogen and the host did not co-evolve, and so the genes should be better. However, we know from experience that these two rye genes, PM8 and 17, broke down very quickly once they were introduced into wheat. And the question was, can we learn something from this? And that was the start of this project. And we first looked at PM8 from rye, and you know that PM8 is on this large 1RS translocation from Petkus rye. And this was then used a lot in wheat breeding because it has all these positive effects on yield and also stem rust resistance. And so it has been really very widely used, even though PM8 as a powdery mildew resistance gene broke down quite rapidly. And at one moment, there were 50% of all CIMIT lines contained this gene. So it was really very widely used, and we wondered what happened on the pathogen side when such a gene is very widely used. And so we set out to molecularly isolate the AVR PM8, and we used GWAS to identify the genetic locus, and that narrowed it to a rather small region. And we then identified the candidate genes in this region, and specifically looked at polymorphic genes between virulent and avirulent pathogens, and this led us then to basically one candidate which we could then verify in the Nicotiana pentamiana assay as AVR PM8. Now, of course, then we went into our large collection of sequenced powdery mildew isolates. In total, we looked at 212 isolates, and looked at the diversity and at the virulence of these isolates on PM8, and we found a total of 17 different gain of virulence alleles in this whole population. And basically, there was only one amino acid which was changed in all this diversity, and that was always at this position 43, and there we find two independent mutations at this position, indicating that this is really a crucial amino acid. But what we found is basically mostly mutations that disrupt the open reading frame of AVR-PM8. So we found mutations in the start codon. You can see here basically all three nucleotides. We found mutants in the start codon. We found 3 of the 4 positions in the splice site were affected in certain mutants, and we had some stop codons also in some of the mutants. So what we think that here we see in the diversity analysis that this is almost a saturation mutagenesis that nature did in this gene, providing us with all different variants that can lose recognition. And when we now look at that geographically, we found that this one haplovariant F43Y, this is present worldwide. This is this white variant, and you can see that it's present basically in all the populations that we looked at, with the exception of Japan and of Australia. And this means that probably virulence was already around globally. This virulent variant was around globally even before the Rye gene was introgressed into wheat. So basically the gene was doomed from the beginning because there was standing genetic variation for virulence. And one could have easily predicted that PM8 would not be functional for a very long time if we would have had this knowledge already then. The other virulence alleles that we found are indicated here, and we find them locally only, and this indicates that they are recent events which evolved only once PM8 was brought into the cultivation in that particular region. So this was the story of PM8, and we found a very similar observation for PM17, the other Rye resistance gene, which was introgressed on a different chromosomal translocation. But basically, it's the same story, and I don't want to show you the data here. This is published work on the AVR PM17 and its evolution and global distribution. Now, what I want to describe now is our work on mutagenesis of powdery mildew to find aberrance genes. Because when we did all this work that I just described, we realized that it would be very good to have an additional method to identify aberrance genes in addition to GWAS and mapping and map-based cloning approaches. And so we basically used a method which was described by Ralph Pansruga's group. It's based on UV mutagenesis. You mutagenize in 3 consecutive rounds your mildew growing on leaf segments, and then you multiply that on susceptible lines, and then you select those which can grow on resistant lines, and so that they have lost the avirulence gene. And then we used whole genome sequencing to call SNP variants, to do coverage analysis, to detect larger deletions, and also to detect transposable element insertions in the mutated progeny. And this has turned out to be a very nice method to get to genes. And this is work that was mostly done by Zoe Benasconi, now postdoc in the group, and Ursin Stirnemann, a technician. So they have been very patient to identify all these mutants that I will now describe. So first, I would like to describe what we did on PM3B and PM3C. And as I already said, these two NLR immune receptor variants, they detect or they recognize the same effector, but PM3B has an extended, an expanded resistance spectrum. And we know that this expanded resistance spectrum is because of a differential reaction to a suppressor gene in powdery mildew. I don't want to go into this here, but we know that this suppressor exists because we can detect it, and we have isolated it at the molecular level based on genetic analysis. So we made the mutagenesis of PM3B, and basically what we found is all mutants on PM3B were also mutants on PM3C, and this nicely confirmed what we already knew from earlier work. And when we then looked at the mutants, one of them had a stop codon, the other one had a large deletion, and very interestingly, 4 of the 6 identified mutants had a transposon insertion upstream of the gene. And when we then looked at the expression level of these insertions, you can see the result here. BC2, that's the large deletion. So there is, of course, there is no expression of this effector gene, but then these are the transposon insertions here, and they have a much lower expression level due to this transposon insertion, and this probably then results in the loss of recognition and in virulence on PM3B and C. So this was very encouraging, and we then moved on into something new, and to the pair of PM3A and F immune receptors, and which also recognize the same gene. But we know PM3A has an expanded resistance spectrum, so it must recognize something in addition. And we set out to identify this AVRX, which is unknown, and we wanted to use this mutagenesis. So we did a two-step mutagenesis protocol. We first identified mutants on PM3F, and you can see here this 3F1 mutant is now growing on PM3F, but it does not grow on PM3A. So we looked at these mutants, and we found several of them, but one of them was a large deletion in the region of the known AVR. And we then used this mutant for a second round of mutagenesis to identify mutants that were not only virulent on 3F, but also in addition virulent on 3A. And we actually found such mutants that would now also grow on PM3A. The surprise came when we looked at these mutants, because we identified a total of 6, and 3 of them had mutations in a gene which we call BGT646. And this is not an effector. It's an unknown gene, which we have here, these 3 mutations. And we don't really know what exactly is the function of the gene, but obviously the gene is necessary for virulence on PM3A, and no, for avirulence on PM3A, I should say. And when we then looked in a bit more detail what these double mutants would actually reveal, we found that of course, they didn't express AVR PM3AF, the known effector, because we had a deletion after the first round of mutagenesis. But when we then looked at other effectors, we saw that some of them showed also much lower expression. But there were also known AVRs which were not affected. And our hypothesis at the moment is that this this gene that we identified, this BGT646, is a transcriptional regulator, direct or indirect, which controls the expression of a subset of effector genes. Certainly not all effector genes, but a subset of effector genes. And among them, the additional component recognized by PM3A. But when we then tested this mutant on a wider range of host lines, we found that this mutant also gained virulence on a totally different resistance gene, and this is on WDK4. In Aguilops Tauschi lines, resistance was lost and virulence was gained, and I will come back to that. So obviously this BGT646 gene not only controls the avirulence gene expression for the PM3A, but also for WDK4. And this is, of course, very interesting observation, which I will come back to later. So in terms of PM3A, we still miss the second recognized component, but we found by mutagenesis this gene which is involved in gene expression, and this specifically affects resistance to PM3A, not to other PM genes, with the exception of this WDK4. So only a small subset of effector genes seems to be not expressed in the absence of this pathogen gene. Now, as you might know, the Brande Wolf has in this perspective paper has made a clear statement that we should now, with the better molecular tools in wheat genomics, we should really create an atlas of wheat disease resistance genes, and that we try to identify at the molecular level all the genes. And we think that we should do the same for avirulence genes. And this has then led to what we call the avirulence gene depletion assay, a much faster way to get to avirulence genes than with the classical method. And I want to briefly describe you this approach. You have an avirulent and a virulent isolate. You make a cross, which you can easily do in powdery mildew, and then you have this haploid F1 population, and you make a non-selected bulk. You multiply this population on a susceptible wheat line, and you make a selection on a resistant wheat line, and then you basically sequence these two bulks, selected and non-selected, and you look for genomic loci which have an allelic depletion. Here there would be a depletion in the AVR gene locus, and this then identifies immediately the genetic location of the avirulence gene. And so we used that approach to test first on a gene, on the PM60 genes, which was cloned from Triticum urartu a few years ago, and we used transgenic lines with and without this resistance gene, and then we made this depletion assay, and this identified one single genetic locus which was depleted, and then we looked at the candidate genes, and basically there was one candidate gene which was clearly the best candidate. It was present in duplicate in 2 copies, and when we then tested in the Nicotiana assay these both copies together with the PM60 immune receptor, we found hypersensitive response, which you can also see here. So demonstrating that both these copies, which are very, very closely related, can act as avirulence gene for PM60. And this again, this AVR PM60 has this RNase fold predicted structure. When we then went to the global population, we saw that there were isolates with 1 copy, with 2 copies, 3 and 4 copies, and this correlated very nicely with phenotype, and only those isolates with no copy of this AVR-PM60 gene were virulent, as you can see here. And when we then looked in the at the geographic location where these isolates are found, we found 2 in China and 11 in the United States. And this would immediately suggest that PM60 is not recommended for use in the United States because there is already virulence around. And this comes as an immediate recommendation based on these data. So this is the PM60 story. This showed that this was really within 3 months we could get to the aberrance gene for this PM60 immune receptor. And we then wanted to apply this to the last PM3 allele that we never found the aberrance gene. This is PM3E. And PM3E is a very good gene. It confers near complete resistance in a global diversity panel. We had to look very, very hard to identify a virulent isolate for a very long time. We never managed with UV mutagenesis to produce gain of virulence mutants. And then once we had identified an isolate from China, which was actually virulent on PM3E, we wanted to move on. And basically, we were this was really a big question to us, that the PM3D and E immune receptors differ only at 3 amino acid positions, and we had cloned the AVR PM3D, but this was not recognized by PM3E, and we really wondered what was recognized by this very, very closely related immune receptor. So we used this aberrance depletion assay once we had this Chinese isolate with virulence, and then at least in one cross we identified 2 genetic loci which were depleted in the progeny, and that was then really the breakthrough that we were hoping for. We started to look at both these loci, and very surprisingly, both these loci contain an active aberrance gene, as it is shown here in the Nicotiana assay. You can see here locus 1 has a gene which is recognized, and locus 2 has also a gene. And the locus 2 encodes mildew protein, which has this classical RNase fold structure, but locus 1 has the aberrance protein looks very, very different. And it's actually a much larger protein and something completely new and very surprising. It was this gene, or this AVR, belongs to a large family which is called cluster 40, and this is highly expanded in Plumeria. And if you look in a study from the Krasileva group 1 year ago, where they did fungal effector structure modeling, they found that, of course, there is this RNase-like structure which is expanded. This gene family is expanded very much, but then there is this cluster 40, which is also highly specific for Plumeria graminis and highly expanded. And now we have, for the first time, a function for one of these effectors that is just one member of many others, and as this is just a predicted structure, we don't really know the real structure. This is AlphaFold, and of course this is now really a new discovery that we want to follow up, because it's the first time that it's not an RNase-like structure that is encoded by an AVR. So to conclude this part, we have learned about natural diversity that reveals evolutionary history and can be used in predictive breeding. That's the work on these introgressions from Rai. We have identified a regulatory gene which controls at least 2 resistance interaction. We have this new aberrance depletion approach for rapid identification, and we have a new aberrance protein. And very surprisingly, this PM3E immune receptor has a dual recognition specificity, which obviously makes it a better gene in agricultural use. Now, to just conclude, maybe in the last 5 minutes or so, I want to describe to you our recent work on non-NLR race-specific immune receptors. And as I said at the beginning, from these 17 genes isolated at the moment, there are 5 do not encode NLRs. And one of them is the PM4 gene, and the other one is the WTK4 tandem kinase gene. And I want to discuss with you now our state of the work on the identification of the specific virulence factors recognized by these 2 non-classical immune receptors. And to start with PM4, we have cloned this gene 3 years ago, and it encodes a novel type of protein, and it actually encodes 2 different splice variants, and we know that both these splice variants are needed for resistance. And these 2 splice variants encode high chimeric proteins with a kinase domain and the C2 domain in both cases, and the splice variant 2 has in addition a transmembrane protein. And this structure here is similar to multiple C2 transmembrane proteins which have been described in Arabidopsis, but in Arabidopsis the genes do not contain these kinase domains. So this is something very new, and of course it becomes even more interesting because Paul Nicholson and colleagues in the John Innes Institute have found that the PM4 gene does not only confer resistance to powdery mildews, but also in a race-specific way to wheat blast. And this work has also been published just recently. And with our transgenic lines, you can see that overexpressors of PM4B show a nice resistance. And if you have strong expression, you have actually also protection of the spike against wheat blast. So PM4 is really a very interesting gene. And of course, it's relevant to ask what is recognized from powdery mildew to induce then resistance. So we that's basically the question: what is recognized by these 2 non-conventional resistance genes? And we used again now mutagenesis to identify mutants, and you can see here that we identified mutants which can now grow on PM4A and B lines, and in total we isolated 6 of these mutants, and they were all mutated in the same gene. And this gene Zoe calls TUF, and this TUF gene is a chimeric gene between a typical RNase-like effector, which is at N-terminal domain, so it's a bit more than 100 amino acids, but this is then fused to a very complex domain, which he calls antigen-like C-terminus, and there are a few subdomains predicted, but it's really not clear what this second domain is doing. And the mutants that Zoe identified are indicated here. These are mostly stop codon mutations, a frame shift, and only 1 amino acid change. And this protein has a length, so it's much longer than the usual effectors, but it has a signal peptide. It's highly expressed. So it's a new type of chimeric avirulence protein, which is recognized by PM4. So again, something novel from the pathogen side. And the second non-conventional non-conventional immune receptor, race-specific immune receptor, comes from the work that was done in the Open Wild Wheat Consortium, which was done under the leadership of Brande Wolf, and we used the Agrops Tauschi diversity panel to look also for powdery mildew resistance, and this has then resulted in this isolation of WTK4. This was described 2 years ago in a paper, and so we wanted to know what is recognized by this tandem kinase, which is encoded by WTK4. It's a race-specific resistance, and we have recently found, which we overlooked at the beginning, is that it also includes a heavy metal-associated integrated domain. And if you look at the predicted protein structure, this is this HMA, heavy metal associated domain. And you might know from work on NLR immune receptor that some NLR immune receptor contain an integrated domain, or HMA domain. These are mostly rice resistance protein. And here we find this domain now also in this WTK4. Now we used biparental mapping to identify the AVR in a cross between aberrant and virulent isolate, and we found one locus on chromosome 5 with only 2 polymorphic effectors, so very few candidates. These are the 2 candidates we have. And then we went on and used a second approach to get to the aberrance gene for WTK4. We used mutagenesis, and we found 2 types of mutants. Again, as I mentioned before, that these mutations in PGT646, which seems to be a regulator, can also result in loss of expression of this PGT55150 AVR. And then we also find mutants directly in this AVR as, so in the same gene that we have predicted from genetic mapping. So we are still in the work of functional validation, but I think that there are this mutagenesis and genetic mapping both gives very strong evidence that this is the AVR. And the multimer modeling of the WTK4 tandem kinase protein together with this candidate from powdery mildew would predict that there is a strong interaction between the 2, and mostly between the aberrance gene and the HMA. So that would be very similar to the NLR interaction with these rice proteins. Now, the prediction is that this would be the most important interaction surface, and threonine 30 would be a key amino acid. And when we now look at this position, we find in one of the mutants, this has only 1 amino acid polymorphism, and it's exactly at this threonine 30 position. This results in gain of virulence. And then also a natural variant in the isolate 2 and 12 has this same mutation. So that gives us really good confidence that this protein from mildew is the protein recognized by the tandem kinase in the resistance interaction. So with this, I want to conclude. I have 2 short summary slides. First, on these non-NLRs, we have this MCTP domain protein PM4, the tandem kinase, and we have for both of them more or less confirmed aberrance gene. For EM4, I think we have confirmed it. For WTK4, we have good evidence, but there is still some functional work to be done. And to summarize the mutagenesis work, I think we have a very good mutagenesis pipeline to identify AVRs for all kind of immune receptors, and we can create double or triple mutants. And so I think based on this work from Ralf Panstruga's group, this is really an expansion of the toolbox for molecular work on mildew. And with this, I would like to conclude. I thank many people over the many years who have contributed to the work, many collaborators globally where we have interacted with, and of course the funders over many years for our work, which I hope I could give you some indication of what we did in the last year on the pathogen side. Thank you very much.